There's hecklers in the front row. Please disregard. Good morning, everyone. I'm Ann Cooney. I'm a social worker with ALS United Mid-Atlantic, and I have the honor of introducing Dr. Colin Quinn today. Dr. Quinn is the director of the Corporal Michael J. Crescenz VA Medical Center ALS Interdisciplinary Clinic. As many of you know, military veterans are twice as likely to develop ALS as opposed to the general population, which makes ALS a service-connected disease. Many veterans with ALS travel great distances for appointments with Dr. Quinn and his team because of their excellent reputation. Dr. Quinn is also an associate professor of clinical neurology at the University of Pennsylvania, where he serves as the director of neuromuscular clinical trials and the associate director of the UPenn MDA clinic. That's a lot. We are delighted that Dr. Quinn is presenting on the topic of making sense of antisense, genetic therapy approaches to familial and sporadic ALS. This is a topic of great interest even beyond the ALS community, with some recent exciting breakthroughs in genetic therapies, including tofersen, which was approved to treat a genetic form of ALS in 2023. Dr. Quinn, welcome to Help and Hope.
All right, mic levels okay? You can hear me okay? So first of all, thank you very much for having me. I really actually love doing talks for patients, partly because it's a great way to figure out if you actually know what you're talking about. Because if I can't explain these things to you, I probably don't know what I'm talking about. So that being said, I'm not here to give you PTSD about biology class. We're not going to try to turn you into a PhD candidate in a half an hour. There's going to be some scary graphs and charts and things that make you think you don't know what I'm talking about. Don't worry, we're going to talk through it. If you have a question, just raise your hand. Don't worry. I have to put data up here, partially so you understand that data is being generated, and partially just because this stuff is complex and we have to talk about it in such a way. Why talk about antisense? It sounds strange to begin with. Well, you know, it's honestly, I think a lot of you know, been a rough couple of months in terms of ALS therapeutics. And I think that these genetic therapies are actually a bright spot and something we can really focus on. It's not the only bright spot, but it is a bright spot that is confusing and tough to talk about. So it's a good opportunity to try to review it and learn something together. So I always just give my disclosures. Some of the things I talk about, I do advisory boards for Biogen and Sarepta and other people who are involved in genetic therapies. I don't think you'll see any bias within the talk, but I just like to let people know that. So what are we trying to do? Well, I probably don't have to tell you all too much about ALS, but I'm going to just do a quick ALS review that's relevant. And then talk a little bit about genetics, because we have to learn just a little bit of biology so that we can have a framework to talk about these therapies. We're going to talk about ASOs, antisense oligonucleotides. That's where the antisense comes from. And then how are they used to treat familial ALS? And then most people don't have familial ALS, so we want to talk about, can they be used in sporadic ALS? I made this slide years ago for a senior citizen science group, which was awesome to talk to. They were just such fun. But I made this slide just partially to show people kind of the journey that physicians go through to kind of land where they are. I've bounced around lots of places to learn the things that I know. And I also put this picture of Bob Brown, who is a huge figure in ALS and actually discovered the SOD1 gene that we're going to be talking about. And he's actually discovered lots of other genes. And I had the opportunity after fellowship to he just called me up and said, basically my wife's fellowship was a couple years longer than mine. And he basically said, I hear you have nothing to do. You want to just come here and hang out with me for a couple years? And who would say no to that? So I worked with Bob for a couple years, and he really, you meet certain people who you realize they get a lot done because they're smart, but it's really how they're positive people who energize other people and inspire other people to do things. And he certainly did that for me. And I've been at Penn for about 10 years, back at Penn. I was a resident at Penn and back at Penn for 10 years. All right, so we always have to have a history slide. I don't know why. I like it because it kind of helps ground us as to where we came from. So ALS was originally, it was not originally discovered. This guy Charcot, this French neurologist, his success was naming it. He gave it a name. It's called amyotrophic lateral sclerosis. Why is it called that? It's such a weird name. Amyotrophy is just muscle atrophy, and what Charcot is actually really famous for is making pathological connections to what he saw in clinic. So he did autopsies on people, and he looked at their spinal cord, and in that bottom right, that's where the motor tracks run. And if you run a probe over that, it's scarred, and so it's sclerosed, it's hard. So the lateral sclerosis is the lateral portion, the outside portion of the spinal cord is hardened. So that's kind of his contribution. He also described the disease, and you'll notice a couple things. What he describes looks a lot like what ALS looks like, right? And unfortunately, the prognosis, although I'm always concerned when I put an average up here, because we all know that none of you who have ALS are an average, right? You are an individual, and you don't follow these average rules. But if we just look at the average prognosis, it actually hasn't changed since the time of Charcot, which is pretty devastating. He said it wasn't hereditary, and he was mostly right, but not totally right. And he gave this pathological description that was how we got this name that seems a little unwieldy. So I would call ALS a common uncommon disease, like the type of disease where once you see it, you can't unsee it. It's everywhere. You're going to know someone, even if you aren't in this room, you're going to know someone who has ALS. And the most significant risk factor, which we still haven't been able to do anything about, is aging. We can't stop aging, and it turns out aging puts us at risk for all sorts of things, including ALS. And again, Charcot said it's not genetic, but we know about 10 to 15% of cases are genetic. There are environmental risk factors, and of course, as was mentioned, I'm the director of the ALS clinic at the VA, and so I have to mention that it's a service-connected illness because there was evidence to suggest that veterans are about 1.5 to 2 times the civilian population increased risk for having ALS. All right, so this is what everybody wants to know. What causes ALS? This slide, don't worry, right? I'm just trying to show you that we don't know. We don't know. It would be a lot simpler. I think we've moved a little bit beyond the blind man and the elephant, in which we're all staring at our individual parts. But one of the themes is that it seems as though a huge problem in ALS is that we have these toxic RNA. Don't worry if you don't know what RNA is, because that's coming in like 2 slides, and toxic proteins that build up within the cell. And those seem like they could be good targets for therapeutics. So one of the things, even though sometimes it looks like, okay, that slide's so complicated, we obviously just don't know what causes ALS, and maybe there are multiple causes, and it's very complicated. But this explosion in our understanding of the genetics, and these are all genes that have been connected to ALS. And what you see is that we start back in 1993 with SOD1, and then it's quiet. We don't find other genes for a really long time. But then you see by the late knots, we're exploding, we're going up. And what on the right, again, no way you're going to know what that graph means, but what those colors represent is trying to connect different biological processes, trying to see themes between these genes, where clearly somehow these genes are important in keeping motor neurons alive. And if we can understand how they're connected, we can understand a lot about ALS. All right, so this is our biology lesson. This is all we need to know just to be armed up for this discussion, okay? So when we think of our genetic code, I mean, it is a miracle. It is a miracle that these 4 little code letters, so these cytosine, guanine, adenine, thymine, these are chemicals that basically encode everything you need to know to make you, okay? And in DNA, they're paired with each other. DNA is this kind of ladder structure that's been twisted, and each of these cytosines is paired with a guanine, and adenine is paired with thymine, and you can see that builds this ladder structure. So these little colors are consistent with these chemicals. That's the coding. That's a genetic code. So DNA is a ladder structure where you have what we call a sense and antisense strand that meet up with each other. And then RNA is this middleman product you have to make to get to protein. Protein is the goal. That's what we're encoding in our genetics. We're encoding proteins. So it goes DNA, double-stranded structure, RNA, single-stranded structure to protein. And this is kind of that process laid out. So the DNA sits in the brain of the cell, the nucleus. It gets unwound and transcribed into an RNA, which is single-stranded. This is important because we're going to target that RNA. RNA actually goes through a little editing process. It has a first draft called pre-RNA, and then that gets edited to messenger RNA. That leaves the brain of the cell, the nucleus, goes out and gets translated into protein. But this is our target, these single-stranded RNA structures. So DNA, RNA, protein. Our target is RNA. Why is RNA our target? Well, people have won Nobel Prizes trying to explain why RNA is our target, right? So for a long time, it was thought, okay, we have a genetic code. And how we control our genetics is turning these genes on and off. That's how we alter what's being expressed and what's not being expressed. And then in the 70s, somebody said, well, you know, DNA's in there too. Maybe RNA's in there too. Maybe we do some regulation through RNA. It's only a little bit of work on that. And then in the 80s, and then these two gentlemen, Fire and Mello, in the 1990s really broke things open, and they won a Nobel Prize for this, looking at how if you give this worm, they won a Nobel Prize for making a worm twitch. Essentially, that's what the prize is for. So they gave these worms double-stranded RNA, and it turned out that when they did that, it knocked down the protein that was supposed to be developed by that RNA. There's an entire biological process within the body. When it sees double-stranded RNA, it targets that RNA. It can target that RNA depending on how you do it for degradation. So you can control how much protein is produced by giving RNA to somebody. And that's essentially what ASOs are. So ASOs, what's in a name? Okay, so antisense just means, again, when we were back here, we had, you can see, right? So we had two strands, sense and antisense, right? RNA just has this sense strand that you've made. What you're doing is you're creating an antisense strand that matches the sense strand. So as I said, those four letters, the letters are a little different in RNA, but don't worry about it. They match up with each other. So you can just create so if you see a G, you can give a C. You can just basically match that RNA. And you only need a few of them. That's why it's called oligo. It's just a few of those to match. You don't need it to be a very long strand. And nucleotide is just the letters that make up the code. So what you're doing with an ASO, an antisense oligonucleotide, is you're creating essentially a synthetic, we call it a genetic product because it can actually be kind of DNA or RNA, but you're creating a synthetic strand that's going to match some target RNA. So there's two approaches that you can use. So the approach that they were talking about when they won the Nobel Prize was trying to get rid of a protein. We call that knockdown. And we're going to talk about that first. So that's reducing a protein you don't want. The other approach is you can actually enhance a protein you do want, which is kind of cool. So you can go in either direction depending on what your approach is. So we're going to take knockdown first, okay? So let's just review real quick. In a regular time here, we have DNA, we make messenger RNA, and that gets translated to protein. In, let's say, ALS and in certain ALS conditions, you have mutated RNA, and you might get a bad protein that you don't want. Like a lot of bad proteins clump up, they clog the cell, they don't do what they're supposed to do. So instead, you want to make an ASO that attaches to that RNA and targets it for degradation. So you don't get the protein you don't want. So we have a success story already using this approach, right? This is SOD1 ALS, right? So SOD1 ALS, as we saw in that graph, was the first gene discovered to cause ALS. And when it was discovered, it was so exciting. It's like, oh, we got this licked. Now we know Now we understand SOD1, it does this protein does these things, and it must be the loss of function of SOD1, and that's why people get this disease. Well, that all turned out to be, if not completely wrong, just mostly wrong. So SOD1 accounts for 20% of familial ALS and 2% of ALS overall, so it's a kind of a small target. It's usually dominantly inherited. So I made this so small because I don't want anyone to even try to read it. Just recognize the green is good. This is when your SOD1 is normal, and the red is bad. When you have mutated SOD1, it tends to clump up and aggregate within the cell and kind of wreak havoc in multiple ways. You can see there's arrows going all over the place. Lots of bad things happen when you have this mutant SOD1. So, all right, we want to target that. We want to say we don't want to produce this toxic protein, SOD1. How are we going to do that? Well, here's the basic science approach, right? So this is way back in 2006. Keep in mind, this drug is approved last year, 2023. So think of all the work that went into that, right? So here is some poor postgraduate student maybe, using all these different combinations of those letters to build an ASO, right? So it's basically they're trying to say, where can we match that SOD1 protein so that we can do the best job at reducing it? Many rats died in the process of trying to target their SOD1, and they pick a winner. They pick this one, and then they go on and say, hey, actually when we give that, it does actually help these SOD1 mice live longer. And as much as we love mice, the goal, though, is not to save mice. It's to save people, right? So just to talk about ALS trials, we kind of have to just say these are the main outcomes. A lot of you know this already, right? So a lot of you in clinic probably have gone over this survey of questions where we're talking about, is your speech different? Are you having trouble swallowing? Well, that's the ALS-FRS, and it's the main outcome in many ALS trials. And then we also look at how strong is your breathing. So how many liters of air can you blow out in a single breath? So those are helpful to know because a lot of the graphs are going to show that. So this is the first crack at it. This is Tim Miller, who's at St. Louis, the picture on the left. And then on the right, his name is Toby Ferguson. He's a former Penn resident and fellow. And you might notice that Toby Ferguson sounds a lot like Tofersen. I don't think that's a coincidence. He worked at Biogen. They deny it. They say it had nothing to do with that. I highly doubt that is true. So what you see on the graph on the left is just different doses. We have to find the right dose. So you've spent all the work trying to find what you think is the right sequence, and now you have to give the right dose. And it turns out you can give a lot of it, because what you'll notice is that the best suppression comes from the highest dose, which is the dose we'll land on, right? But again, that's just measuring, was I successful in knocking down the protein? Did I knock down SOD1? And you did, and it was better if you gave more. So give more, right? But knocking down SOD1 protein doesn't matter if people don't do well, right? If people don't functionally do better. So this is a very small, see, N of 12. That means 12 people in the placebo group. The placebo is blue. Tofersen is orange. This is a very small trial. This is just trying to get this was what was, the left-hand side is what they really care about in this case, because they really want to know, are we even doing what we think we're doing? The right-hand side is like, good to know, but not enough people to actually prove anything. But there is a suggestion here that the orange lines are going straight across and that the blue lines are dipping, meaning that their ALS-FRS, their score on that survey is going down, whereas the Tofersen patients seem to be doing better. So this was very exciting. So that leads to a larger trial, right? And at the highest dose, that makes sense. And for every 2 patients that got Tofersen, 1 person got placebo in this trial. We were part of this trial. And that survey of symptoms was the primary outcome. They also used biomarkers. Biomarkers are a way that we can sometimes detect changes earlier and tell whether or not we're hitting targets. So we looked at spinal fluid SOD1 protein levels So you want to know, are you engaging your target? Is your ASO, which is supposed to block the RNA from being translated into SOD1 protein, is it actually doing that or not? And then they also measured neurofilament light chain. Neurofilament light chain is a non-specific marker of neuron injury. And the thought is that if you're having less neuron injury, your neurofilament light chain level, both in the blood and in the spinal fluid, will go down. So I was excited to participate in this trial, and it is the first time that I have ever been in a trial where I'm like, I can tell who's on drug. I can't ever tell who's on drug. And people always ask me, am I on drug? I'm like, no, I don't know, and I'm not supposed to know, right? But in this case, I really felt like, I feel like I can tell you're on drug, because the effect size seemed large enough, meaning that this person who I was seeing was not getting worse when they were clearly getting much worse before I saw them. But here's what's scary. So on the left are the biomarkers. This graph requires some explanation because there's a lot going on here, right? So the solid lines the red are people who are progressing quickly. We kind of divided the groups into people who are progressing quickly versus people who are progressing slower in blue. And the dashed lines represent placebo, and the solid lines represent tofersen. What you'll notice is the biomarkers look great. We reduce SOD1, and we're reducing neurofilament by a lot. By a lot. That is a significant reduction in, I mean, it's 50% here, and actually it turns out it's more when we look for a longer period of time. That is a very substantial reduction in that marker of neuron injury. But when we look at the ALS-FRS and the vital capacity, they're separating. The dashed lines and the solid lines are separating, and in a direction that tofersen is higher than the placebo, but they're not, those error bars are overlapping. They're not separated enough. So that was at 6 months, and it was incredibly disappointing because again, I've never been sure that a drug works, but I was pretty sure this drug worked, and so to get those results back was pretty heartbreaking. Fortunately, they were smart. They were dumb about some things and smart about other things. So the trial didn't just stop at 6 months and send everybody on their way. What they did is they did what's called an open label extension. Open label extension means, you're blinded in a randomized controlled trial, right? You can't see what's on the label in this theoretical pill bottle. You can't see what's on the label. You just take the pill out and put it in your mouth. In this case, the label has been opened. You can see that it's tofersen. Everybody's on drug. At 6 months, which is represented by this black line, everybody goes on drug. Now, what's interesting about doing that is everybody's on drug, but you're still going to try to do your comparisons to the people who are started on placebo. So it actually handicaps your ability to see a difference because they're no longer on placebo, they're on drug. So in theory, they could catch up to the people who are on drug. But what we saw was that those people who were on placebo, the dashed lines, they do start leveling off and doing better, but we're starting to see separation, pretty clear separation between, on the ALS-FRS, although these error bars are still overlapping, but in the vital capacity, we see statistically significant separation. And we continue to see suppression of this mutant SOD1, or just SOD1 in general, and the neurofilament light chain. So what was the FDA going to do here? Because the main outcome was missed, but you have a drug that people are pretty convinced works. Well, they were convinced enough that that neurofilament level change was too hard to ignore, and then the open label data really seemed to support that the drug worked. So the drug was approved, and we've been giving it commercially for about a year. I was just saying that I give it right in clinic. It's not super convenient. You still have to get a lumbar puncture every month. But I've had one gentleman, actually the gentleman in the picture, he's public. I mean, he allowed us to take this picture. He's received, I think, 48 treatments, and if you map that out by monthly treatment, he's doing pretty well in terms of how long he's been dealing with the disease, still teaching high school, still doing yard work, still walking into clinic. He's done really, really well. So a big question we get is, okay, that's interesting. When do you treat? Because not everyone responded to this therapy, and I think that hasn't been analyzed enough. But what seems to be clear is that earlier is better. Well, how about before you even have symptoms? How about we not get weak at all? Wouldn't that be nice? And there's an entire trial that's going to take a really long time to complete called the ATLAS trial that's looking at that. And what they're doing is they're looking, because if you just start treating someone and they weren't going to develop ALS for another 20 years, your trial is going to have to be really, really long to figure out whether or not you've made any difference. But what they're looking at is that neurofilament light chain, that marker of neuron injury. They're looking at that. It seems like that goes up before people actually develop weakness. So you start to injure, it kind of makes sense. You start to injure neurons, you release this neurofilament light chain, and then eventually end up with weakness. So they're kind of monitoring that neurofilament light chain, and if it goes up, they start treating people. It will take a long time to get those results. And then, again, do not read this slide. But the point is just that ASOs are not very convenient to receive. There is a way to deliver this by gene therapy in which you infect someone with a virus and you basically have them run off RNA that will act like this antisense RNA interference and also knock down SOD1. So we've been working for actually 2 years with a company that's changed hands to get this trial running, where it's an AAV targeted against SOD1. And the hard part is actually going to be finding people for that trial now, because if tofersen's really effective, it's actually hard to tell people, actually, do you want to do this thing that we don't know if it works or not? So that's complicated. All right, so there's a sense of like, okay, that's even before the tofersen was approved last year, it kind of seemed like this made sense as an approach. And there's a fair number of familial forms of ALS. So let's just fire up the engines and treat all of these this way. And the most common is this expansion in chromosome 9 called C9orf72. I apologize for the name. I did not make it, but it explains about 40% of ALS. That's if you're in the US or Europe. In Asia, it's actually very little. But here, it's a significant contributor to familial ALS. It also has toxic proteins and toxic RNA that you don't want. So seems like a perfect target. It seems like a slam dunk. But unfortunately, it hasn't been that easy. So there were 2 trials that were stopped. Biogen, who made tofersen, tried to make this C9 antisense. And in this case, they actually saw the neurofilament going in the wrong direction. The neurofilament went up. And that kind of, I think they felt that they learned a lot from the tofersen trial, that the neurofilament showed them what was going to happen. I don't think that's always true. But I think the learning from that trial caused them to have a very low threshold. If a drug was causing neurofilament to go up, they were not going to keep going with it. And then another company called Waves also stopped a C9 program. I don't think that means that that's done. C9 is more complicated than SOD1 for reasons we don't have to get into, but there are still going to be C9 trials coming. But it definitely, we kind of felt like we were on a roll, and then these couple trials happened. And that's how these things go, unfortunately. It's just when you think you've got something figured out, you have to learn something new. But what most of you probably want to know is, okay, this is really interesting, but we're talking about 2% of ALS, or even if we treated all familial ALS, it's 15% at most. Does this really help me? Well, fortunately, as I showed in that incredibly confusing picture, which now I've made so small you shouldn't even try, right? This is just what an ALS scientist has to keep in mind. I know a lot about the pathology of this disease. And we know that part of the pathology of the disease is we produce RNA and protein that we don't want. And that's true in sporadic disease too. So can't we target something there? So we have to learn a little something about TDP-43, which I'm sure you all woke up this morning and saying, like, man, I really hope I learned about TDP-43 today. But TDP-43 is important. It's actually Penn was really important in discovering TDP-43, and over 90%, like 95% of people with ALS have these TDP-43, this protein accumulation in their neurons. In this little picture here, this is just a stain that shows TDP-43 gathering in the neurons. And TDP-43 is supposed to sit in the nucleus and help process proteins. And what happens in ALS, for reasons we don't understand, is it gets stuck out in the cytoplasm away from the brain of the cell and just aggregates and screws up a whole bunch of stuff. But it's not doing its job, which is handling these RNAs to get processed to protein. So that brings in, I said there were two approaches, right? So the first approach was, let's knock down a protein that we don't want to be there. Well, this is a second approach in which we can say, it looks like the movement from RNA to protein is getting messed up, and we want that protein. That protein does something good, and we want to encourage that protein to be developed. So we just have to recall a little bit from a prior slide, which is we don't just, even though I said DNA, RNA, protein, it doesn't actually go that way. It goes DNA, first draft RNA, final draft RNA, protein. And that's called splicing, when you kind of cut down the RNA to the actual final draft that you want. And with TDP-43, that splicing gets messed up, and you actually end up including parts of the RNA that you hadn't intended to in the final draft, and that results in a protein that has something in it that it shouldn't. And when proteins have something in it that it shouldn't, the body knows that, and it'll get rid of it. So you don't actually get the protein that you wanted. So in this case, we're splice modulating. We're changing how that protein gets edited back to kind of how it was supposed to, so that you get effective protein. So how do we apply that? Because, all right, so TDP-43, it helps with this processing of RNA, but it's the other proteins that aren't being produced that are the problem. How do we know which proteins are important? Well, the fact is we don't know them all. It has an effect on lots of different proteins. But one of the areas of focus has been this protein called stathmin-2. And stathmin-2 is important in neuron growth and maintenance. And when I simplify this diagram, it's still not that simple, so I will admit that. But so when you have normal TDP-43, TDP-43 kind of latches on to this pre-mRNA. This is the first draft RNA, right? TDP-43 kind of is sitting on this portion and not allowing it to be included in the final protein so that you get the protein that you want. You don't want this little red piece in there. And what happens when TDP-43 isn't sitting in the nucleus where it's supposed to be, that part gets included and you don't get stathmin-2. So what you can do is make an ASO that goes after that part of the RNA and attaches to it and says, you're not getting translated, you're not getting through, and then you get functional stathmin-2. So sounds cool in theory, sounds cool in a dish, but we're moving beyond that, right? So there's already a phase 1 trial, I think it's Canada and Europe, that is trying this approach out, where you're targeting and you're trying to make more stathmin-2 to make your neurons healthier. So this is just a summary of all of the current trials. I'm sure it's pretty recent, but I'm sure it's already out of date. And we see SOD1 on there, C9. FUS is another success story, although limited. So FUS is another rare gene that unfortunately often affects young people and progresses very rapidly. And there's a group at Columbia that's developed an antisense therapy for that, and it seems to be quite effective. We are doing a trial in ataxin-2, which is kind of a weird crossover of there's a kind of intermediate expansions of ataxin-2 are associated with ALS, but then it also seems like ataxin-2 may be important in sporadic disease. So that trial has both people with the expansion and people who just have sporadic disease. Stathmin, we talked about, UNC13, these other things. So there's lots of different areas. And I was just pointing out that, it comes back to this, do you want to knock it down or do you want to build it up, right? So some of our strategies are knocking down these proteins and some of them are trying to increase them using just the tools that we've just talked about. Right? So, my hope is that these things, you have some sense of kind of what we're trying to do in each instance. All right, so just want to do a little pros and cons for sporadic ALS. Right? So ASOs seem like they get to target and that they do something, which, got to be honest, in ALS is not that easy to do. So the fact that that has been accomplished is huge. It also seems like we're targeting things that are upstream. A lot of times it feels like, when we're modulating inflammation, a lot of our targets are inflammatory. It kind of feels like the horse is already out of the barn and we're trying to do the best we can, but we're way downstream of the problem. It feels like ASOs are pushing us upstream towards these problems in the cell that are fundamental. The con is that, especially in familial ALS, we know the gene problem. We know we produce this mutant thing. We don't want that. Let's knock it down. The C9 example shows us that it's more complicated than that, but generally that works. In this idea where we want to push up proteins that we're not making enough of, it's hard to pick a winner, right? Obviously, we do this in mice and in addition, all these things, but it's still hard to know whether or not that will translate to having the effect size that we want in people. We want drugs that really, really, really work, right? We don't want you to be on drugs where you feel like, I can't tell if this is doing anything. It's so frustrating. I know how frustrating that is. And we want bigger effect size. And that, my hope is that we're going to hit a winner here, but we might have to deal with some smaller effect sizes first. And the other thing is that protein missplicing, this kind of not producing proteins that you want, is not the whole story. We saw that diagram. It's pretty complicated. That's not the only problem. My hope is it'll have a big effect, but it's not the only problem in ALS neurons. So I really appreciate you all sitting through. I only saw a couple people fall asleep. You did great. You really did do great. I just want to thank all the people that I work with and for being invited to talk with you. I'm happy to take questions if you have questions. Sorry, go ahead.
So whether it's sporadic or familial, does every patient exhibit the wrong proteins and neurofilament increases?
So neurofilament is a marker of neuronal damage. So yes, although what's been interesting is that people can have ALS and their neurofilament is not actually high, which is a little creepy, right? That doesn't make sense with all the things I've said. And then it's also, so SOD1 was our model for years and years and years, right? But it turns out SOD1 pathology is quite different. They don't have TDP43. So I said, 95% of people have this TDP43. Well, SOD1 and FUS, the two kind of genetic targets that we've used, they don't have TDP43. Now, part of that might be that especially for FUS, that's also an RNA processing protein. So you have an RNA processing problem in that setting. So the pathology among sporadic and familial is often similar, but the familial has some of its own stuff going on. Go ahead.
So, congratulations. You did a great job.
Oh, thank you.
Very confusing slides, and I feel like I learned a lot. I just want to ask you, going back to the point at which you described the 6-month, and then I think you called it the open label.
Open label, yeah.
Process. So, do you think that's going to be happening more and more? It seemed like a breakthrough kind of thing, and I'm wondering what you could say about how do researchers recognize when to advocate for that? What were the signs to you that was a worthy thing to request or petition for, however you would describe it?
So I think actually ALS patients did a great job advocating for themselves, and I think that it was just recognized that was going to be a thing from the jump. I also think that because of ALS patients and advocacy, open label has become standard even before tofersen. it has become pretty standard. Not all the time, but most of our trials have a plan for an open label. Because it turns out that it is mutually beneficial. I mean, some of this, I would love to think that it's just like pharma companies are so nice, but some of it I think is that you do learn a lot from just keeping people on the drug. It kind of turns into what we call a delayed start, right? So some people have been on the drug from the beginning and some people have been on the drug, let's say 6 months later, and you're looking at the advantage of starting earlier. So there's real advantages to keeping people on drug. It's expensive, just to, that's true, it's expensive. And it also means, interestingly, for trial sites, trials go on forever. So once we start a trial, we're wedded to, until it doesn't do well, we're wedded to that company for the next however many years it goes on, which if it goes on a long time, that means things are going well, so it's not like I'm going to be upset about it. But it adds complexity to kind of the environment in which we do trials. Go ahead.
Like how effective is the tofersen in terms of quality of life?
Yeah, so I really don't like anecdote in substitutive data, but I'm going to give you an anecdote. So it varies. It varies. And some people do really well and some people haven't done well. And again, I think my hope is that this will be the year where we do a little deeper dive on how do we decide that? How do we figure out who's going to do well and who hasn't? I will say for the gentleman in the picture, this is the anecdote part, right? So, and this is all public. I'm not sharing anything he hasn't shared publicly. He barely qualified for the trial. He actually did not qualify for the trial when he came in because his vital capacity was too low to be in the trial. Now, he was in a thing where the vital capacity had to be pretty high. It had to be 70%. So it's not like it was low. And we had to send a nurse out to his house just to, this was just a quirk of the trial that they let us send someone to his house when he was fully rested to do the vital capacity testing. He had lost a lot of strength in his arm. That is a long time ago. His vital capacity is still in the 50s, which you can imagine once you start to lose vital capacity, that you don't usually slow down like that. He's still teaching high school, still, I'm hesitating because I'm actually not sure if it's middle school or high school, and I think that there's a big difference between those two, but he's still teaching. He still does yard work. It's not like life isn't hard, but I would say he would argue he has quality of life. And it's not just that I think what a lot of people worry about with really is all right is, are you just adding on time to the part of my life that's going to be the hardest, And the goal here is to maintain function. And I do think we can argue that he's maintained function and that other people, so I'll go away from anecdote and go to data, there's, Tim Miller has presented this multiple times, that there are a fair number of people, 20% of people in the trial who actually had increases in their strength, which is pretty wild. You must be pushing on that disease pretty hard to have the space to regrow neurons, to get collateral sprouting and to get strength back. That's got to be something good. I think the mystery is in why doesn't everybody do that well? Why isn't it just so obvious when you look at those graphs that, you just give it 6 months and they separate, He's been on it for, let's say 48 treatments. That would be 4 years, but you subtract 2 months for the induction period, so just under 4 years. Right, so yeah, that's the math I'm talking about. I'm talking about significant atrophy and weakness of one arm and a vital capacity that's not normal and has dropped, and then 4 years later, still above 50, still walking, still doing stuff around the house.
How many patients are in the open label extension at this point?
Yeah, so the open, I don't know how many are left, John. I think it ends in a couple months. So for example, he transitioned, everyone's going to transition to commercial drug in the summer.
And will Biogen continue to gather data?
Well, I'm sure they'd love to continue to gather data because part of their contention is that the overall survival of this population is just going to be substantially longer, right? Like those graphs don't really show survival because it's a tough outcome to, right? It takes a long time. Hopefully it takes a long time, right? So yeah, I have a feeling they are very enthusiastic about keeping everyone into the open label as long as possible. So I suspect there'll still be some follow-up.
So in your estimation, from experience, is tofersen the first great foothold in the search for answers for ALS?
Yeah, I think again, I always look at it as, can I tell? Can I tell? Can the patient tell? And I think riluzole is important, right? I do think if you run riluzole through any database, you will see that people on riluzole do better on average than people who don't. But you can't tell that, and I can't tell that. I can't tell, right? And so it's the first time I've ever felt like, oh, I can tell. I can tell. You're on drug, and you're doing this well because you're on drug. There's, barring miracles, which happen, right? You shouldn't be doing this well at this point. But you have to caveat that with it's in a targeted gene that we know, and there's leaps here to, does that extend out to other forms of familial ALS and sporadic ALS?
So with this ongoing success, because we're still learning about the effect of tofersen, and Biogen is not a small company
Yeah.
in the pharmaceutical industry. Do you think there's a point where other big pharmaceutical companies, they may be already, I don't know.
Yeah, they are. Yeah, other pharmaceuticals jumping into the space.
Well, exactly.
Yeah. Yes, I think that it is definitely an area that companies are interested in, and if you go to a meeting, you'll see a lot of them. There've been, the C9 thing was a setback because I think, there's the point at which everyone thinks it's going to be easy and everyone's jumping on board, and you don't have to know that much. Just make a synthetic RNA that matches this thing and let's go. Obviously they do a little more than that, but yeah, it turned out to be a more nuanced conversation, and that might scare some people. But no, I definitely think there's more interest.
And how far do you think we are from getting away from lumbar puncture and going to AAVs as a route to administer?
Yeah, so we're not far at all from a trial of that, right? The fact that you have to retreat with ASOs is a con, right? Because who wants to get poked in the back every month? Although it does get easier over time, because after a while I just know exactly where, you have a little pink mark and I know where to go.
Good.
Yeah, I've been practicing, I'm getting better. But the issue is, that's a con, but it's also a pro. If something bad is happening, you just don't take it anymore. And bad things do happen with ASOs. I kind of skipped that slide, but ASOs can trigger, you're putting a protein in there that's not really supposed to be in there, or you're putting a thing in there that's not really supposed to be there. So it triggers inflammation, and some people it triggers enough inflammation that they get inflammation of their spinal cord, or they get kind of impairment of the ability of the fluid to flow around their brain and spinal cord. So bad things can happen. If you give an AAV and bad things happen, my sincerest apologies. You can't take it back. We do have strategies for mitigating that, steroids, immunosuppression, because a lot of it's inflammatory, right? But we're still in we don't actually know what to do. There are people who are working on strategies beyond just suppressing the immune system. So some people, this is people at Penn working on switches, where you basically can give a gene therapy that only turns on if you take a certain medicine. So you can shut it on and off, because that's the concern, is that you can't shut it off once you put it in. The downside to that is that you're reducing the efficiency of anytime you put a step like that in between, you reduce the efficiency of what you're trying to do. So I think, it's still pretty wide open. AAV is definitely going to be a thing. We should be starting that trial imminently. The hard part, I think, is going to be finding someone who doesn't want to do tofersen. They originally basically said if you'd ever taken tofersen, you couldn't be in the trial. And that was going to be a problem. So they recognized that, and they fixed that and said there's a washout period now.
So then, there's the damaged microfilaments. So not everybody with ALS has that when you test it.
Yes, most people will have an elevated. So I originally thought, I've gone through a little journey with neurofilament in the last several years, because the perfect scenario, I thought we had the perfect scenario for when neurofilament would be helpful. And that is, we have a gentleman who has familial ALS, not clear what the gene, we were not clear what the gene was, but multiple people in the family who have ALS. He develops ALS. We test. He has a variant of SOD1 that we think is disease-causing, but we're not actually sure. It's a variant of undetermined significance. I thought, well, this is perfect. We'll measure neurofilament. We'll give tofersen. And if it drops, we know he's on the right therapy. And if it doesn't, maybe that SOD1 isn't that relevant, although I still probably would have been tempted to treat him as long as he was doing okay. Turned out his neurofilament was normal. So I was like, well, I can't fix normal. So we've just kind of treated him and see how it goes. The most important thing is whether he's doing well, which he is. So it's a little more fidgety than you would think. It seems so clear. This is a marker of neuron injury. Just measure it and try to reduce it. And if it's high, you have the disease. That's not true. It's much more complicated than that. These things, like many things, work on average. And in individuals, it's a much more nuanced conversation.
Does that marker also show up with other neurological?
Yes. MS has used it for years.
So you couldn't do that if you're unsure of the diagnosis for ALS?
It's actually a great question, and that's, so there are diseases. So multifocal motor neuropathy is an autoimmune disease that affects multiple motor nerves. Sounds a lot like ALS, but it's autoimmune in response to treatment. And sometimes we have a hard time telling between the two. And there are people looking at whether or not you can differentiate the neurofilament light chain levels. The answer is you can, but it's that whole individual versus average problem, right? On average, the ALS population will have a higher neurofilament level than a person who has multifocal motor neuropathy. But an individual person might fall out of range on either side of that. So unfortunately, we still have to do a good exam. We still have to talk to people. We still have to do all these kind of basic things that a lab test just can't cover.
But it's another because you do EMGs and you do
exactly.
to rule things out.
It's a data point. It's something that can be informative, but you really have to not over-rely on it.
I've also heard about stress proteins. I knew somebody was working on a way to measure them. Are those the proteins you're talking about, the ones that get all clumped together?
So TDP-43, when it clumps with these things called stress granules. And when your cells are in a state of stress, they form these clumps, and it basically it goes into emergency mode. We were talking about when your loved one is sick, you're in emergency mode, right? You just deal with what they're dealing with. And a lot of other stuff falls by the wayside. And then sometimes you look up and you think, my God, this room could really use a coat of paint, but we're not doing that right now, It's similar actually within the cell. They form these stress granules and they just say, we're not doing maintenance, we're doing survival mode, right? So lots of proteins that are supposed to be doing things don't get made when you're in survival mode. So that's part of this. That's part of the story too. In this case, TDP-43 is locked up in those stress granules, right? So it's not doing what it's supposed to be doing. But TDP-43, we focus on it because it's a common pathology. It's something we can see. between people who have ALS, but it's not the only issue in those stress granules.
So one final question I think on it is, so those stress granules might be an indicator of ALS as well, but it sounds like they might show up in other diseases where the cells are stressed.
Yeah, if the cells are stressed, you get stress granules. Stress is a colloquial term, but yeah, you get these stress granules and other things too.
Okay, thank you.
Sure.
I learned a lot.
Yeah, we were talking, I gave myself a 70% chance of making sense, so we'll see. I made it, okay, thank you.
We'll do better next.
Yeah, I'll try. I keep trying. It's just like lumbar punctures. Once you do them enough. Okay, well, thank you all very much. I'm happy to take questions if somebody wants to pick up.